Very good evening to all of you. I'm Dr. Vandita Kakkar from University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. And today I'm here to present a very interesting talk on a topic novel THC-Tacro Ointment for Atopic Dermatitis, Synergistic Antioxidant and Anti-Inflammatory effects through nanoparticle delivery. As we all know, nanoparticulate delivery is a very hot area on which all of us we are working to have enhanced bioavailability of any active molecule. So in this investigation, basically, we have worked with atopic dermatitis, which is a chronic relapsing anti-inflammatory skin disorder, and particularly it is having a prevalence of 1 to 30% in different regions of the world. This disorder is mainly prevalent in the pediatric population, in the younger population, as compared to that of the adolescent. The pathogenesis is very unclear, but yes, it involves antioxidant mechanism. There is a lot of oxidative stress which is generated. A lot of inflammation is there. And apart from this, there is immune system which is involved. So currently available therapies, they focus on topical corticosteroids as first-line therapeutics and topical calcineurin inhibitors like tacrolimus, which is named as Protopic in the market, which is approved by US FDA as a second-line product. Apart from this, generally antihistaminics are given for normal pruritus, which happens in children. In lieu of this, we thought of making some nutraceutical based product. We all know curcumin is a pleiotropic molecule, but it is the yellow color of curcumin which limits its bioavailability, and the yellow color limits because of the staining properties of curcumin. It is not mainly involved. We don't prefer it to make an ointment or a hydrogel. So our proposition here was to develop a molecule which is going to be non-staining. It is going to have anti-scarring properties, is very stable. So tetrahydrocurcumin was the molecule which was photostable, non-staining, and had superior antioxidant properties, and even was stable at physiological pH vis-a-vis curcumin, which degrades at physiological pH. So coming to white curcumin, which is also tetrahydrocurcumin, as I referred that it is a very good antioxidant, anti-inflammatory, and has good antimicrobial properties as compared to parent molecule curcumin. We developed this molecule in form of nanoparticle for dermatitis. Clubbing up with tacrolimus was another motive behind, because it is the immune system which is involved in dermatitis. And when we want a complete cure, we want a therapeutic which should cater to all the things: the antioxidant, that is the oxidative stress which is generated, and the inflammation, along with that inflammation, the immune system. So tacrolimus is an T cell drug and acts by inhibiting the production of associated cytokines and suppresses the inflammation. However, there was a problem with this molecule. So when we talk about the market, tacrolimus is available in form of an ointment in two concentration, 0.1% and 0.03%. But there was a black box warning issued by US FDA that 0.1%, if used for prolonged use for dermatitis or any skin disorder like psoriasis, might have an carcinogenic potential. In lieu of this, we thought of producing a formulation which is going to be a combination of nanoparticle of white curcumin, because white curcumin is very poorly soluble in water. So we wanted to enhance the solubility of white curcumin, and then we wanted to club it with tacrolimus at a concentration much below 0.1%, so that we don't have any chances of getting the carcinogenic potential or the chances of any malignancy. So these were the problems as discussed, that the log P value though is good, 2.98, but the molecule exhibits a very poor bioavailability because of the poor solubility, which is only 56 nanogram per ml. So nanotechnology to rescue. We all know that nanotechnology is a boon word. And when we talk of it, we have a range of molecules which are being developed. lipidic nanoparticle, polymeric, liposome, micelles, microemulsion, nanosuspension, dendrimers, phospholipid complexes, and SMIDs. And ours was the lipidic nanoparticle because of the advantages. We didn't use any organic solvent, it's biodegradable, and it could incorporate it has the capability to incorporate the drug with much higher entrapment efficiencies. And because it of the particle size which we are going to attain, it's going to cross across the skin and increased retention on the skin because of the lipidic coating. So it's going to increase the mean residence time and reduce the drug because of the enhanced bioavailability. So the purpose here was to propose a synergistic combination of both of these tacrolimus in a concentration of 0.03% and white curcumin lipidic nanoparticles 2%. So this was the plan of work. The pre-formulation studies very clearly indicated the concentration of phospholipid G to be 0.7%, while the solubility studies in different liquid lipids and surfactant was carried out. These studies were conducted because afterwards we wanted to determine the entrapment efficiency or the drug content. So we wanted to know whether these molecules, they are these tacrolimus and white curcumin, in what molecule, what base should we consider so that we can solubilize them easily. So on the basis of solubility, tween 80, phospholipin at 90G and compritol was selected as surfactant, co-surfactant and lipid respectively. The HPLC method was validated for both THC and tacrolimus and quite good as per the validated ICH guidelines we have for bioanalytical method validation. Both the methods for THC and tacrolimus, they were developed. We could attain all the parameters which were in range. The percentage RST was found to be less than 2%, which is as per in accordance to the ICH guideline Q2R1. Coming to the optimization and development of formulation. So THC LNs, they were experimentally optimized using an experimental design, and the contour plots were made. They were drafted with the optimized concentration. The 3D response plots were portrayed, and the effect of tween 80 and the compritol 888 ATO, that is atomized compritol, was seen. The effect was seen on the particle size, entrapment efficiency, and the total drug content, and based on this, F1 was selected with a concentration of compritol as 7.5%, 16% of tween 80, and we could attain a particle size below 200 nanometer, which was our main target, with 86% entrapment efficiency and 95% total drug content. The method of preparation was very simple, spontaneous method. The purpose of developing this formulation was that we wanted to have a commercial angle. We have applied for this patent, and it was applied in 2018 and it was granted in 2021. And for this, the SLN of white curcumin, even, they have been transferred, the technology has been transferred to industry high-tech formulation in Baddi. With this simple method of microemulsification, we had two phases, the lipidic and the aqueous phase, which were heated at a similar temperature of more than 10 degree above that of the lipidic nanoparticle lipid melt temperature, which is 72 degree. And then this hot microemulsion was transferred into equivalent amount of cold water to form the SLNs, which were then concentrated. And the drug content was determined. After that, they were incorporated into the tacrolimus ointment. So this tacrolimus ointment formula, the base formula, is what we have in the Indian pharmacopoeia. Coming to the characterization part of white curcumin lipidic nanoparticle and the ointment, the particle size, as discussed, was below 200 nanometer, PDI of 0.2, zeta potential minus 15, total drug content 96.60%, and entrapment of 90%. XRD, NMR, they showed that in SLN no sharp peaks were there, which quite, it was evident that they were amorphous in nature and there was no crystalline peak which was observed as seen in C and D. SEM studies showed the morphology of the lipidic nanoparticle of tacrolimus, which were incorporated in the tacrolimus ointment. This was, in fact, a very interesting investigation that we could observe the nanoparticle being present in the ointment, which actually cleared our concept that yes, the nanoparticle, they were quite intact and they are going to go across the skin once applied topically. The pH, the spreadability factor was quite acceptable as per the marketed formulations. Stability studies indicated them to be stable for 2 years at all the temperatures, whether it was 25-degree, 40-degree temperature. So both of them, they showed the lipidic nanoparticle and the tacrolimus ointment, they were quite stable at both the temperatures. Dermatokinetic study showed that THC showed a burst release within 15 minutes, followed by a sustained concentration for 24 hours, and this was attributed to the desorption and the diffusion of the unentrapped white curcumin associated with the interface between the co-surfactant and the surfactant layer, that is the tween 80 and the phospholipon 90G. And it showed 40 times enhanced permeation as compared to the free THC. The in vitro cell line study clearly depicted the efficacy of these produced lipidic nanoparticle, because it wasn't possible that ointment could be tested. So lipidic nanoparticle, they were tested in 3 different cell line and no cytotoxicity was observed in the cells up to concentration of 20 microgram, illustrating the biocompatibility of the treatment group. Thus, it was very clearly concluded that all the ingredients used were quite safe and non-toxic for topical application. Quantitative uptake and qualitative uptake was being observed. These are the fluorescent images of the cellular uptake of the rhodamine 1,2,3 loaded white curcumin lipidic nanoparticle, which were tested in the human cells, that is the fibroblast cells. And they are depicted by the green fluorescence, blue fluorescence, nuclei stained with DAPI, which is the control, and the rhodamine THC LNs. So the HTF and the NHEK cells demonstrated the concentration-dependent uptake, showing increase in the uptake with increase in the THC LN concentration, while the rhodamine LN showed the nanoparticle can be taken up by the cells through the cell membrane. And this green fluorescence clearly depicted, they confirmed the presence of the rhodamine white curcumin LNs, thus showing the importance of having a nanoparticulate-based formulation for topical delivery of these drugs. The quantitative endocytosis pathways was established, all the pathways were explored. And our findings very clearly indicated that white curcumin LN, they followed the clathrin-mediated pathway in HTF and NHEK cells. However, the active energy-dependent endocytosis and micropenocytosis also contributed to the uptake in HTF and NHEK cells. The cell cycle analysis depicted that after 24 hours of treatment with THC LN, blank LNs, and white curcumin per se, there was a decrease in the cell on the G2M and increase in G0, G1 phase. The results were quite similar to that of control, indicating the formulations to be safe. So the final study was the proof of concept study that proved that our formulation is working quite well. So in this, the first one was the in vivo skin distribution using confocal laser scanning microscopy, which very clearly depicted the cutaneous uptake and distribution of LNs in the skin, which can be seen by the green fluorescence here. So the LNs containing the fluorescence dye per se and the ointment clearly depicted deeper penetration of the fluorescent marker as compared to that of the free dye in the ointment. The second proof of concept study was the in vivo pharmacodynamic study, which was conducted in an animal model, which was induced using DNCB, that is dinitrochlorobenzene, which is a standard molecule used to, induce atopic dermatitis in animals. And the results clearly showed that the in vivo pharmacodynamic evaluation for assessing effectiveness against the atopic dermatitis model of inflammation revealed the enhanced bioactivity of white curcumin post incorporation into lipidic nanoparticle and its combination with tacrolimus. It can be very, very clearly depicted. We can see tacrose is the marketed molecule, and E, this can be clearly seen, that on day 22, very clear skin was observed, and this white curcumin, because it is a depigmenting agent. So no scar was left after treatment with white curcumin lipidic nanoparticles, and even the white curcumin topical ointment, tacrolimus and white curcumin lipidic ointment. So this could be seen by scoring. The scorads can be seen, and just a minute. So sorry. Coming to the scorad, which is mainly used for the atopic dermatitis, it could be seen that this scorad, when compared to the TTO, if we compare all the formulation, the first one is the THC-Tacro Ointment, we can see that the scorad was the least in our case as compared to all the other treatments, thus giving a proof that the formulation is very effective in treating dermatitis. Biochemistry and hematology. So the TTO treatment related hematology and biochemistry showed no harmful effects, and the control group showed no significant changes in the biochemical assay. All the values, they were within the normal range for all the biochemistry and the hematology parameters which were being tested. So these were the parameters which are the antioxidant markers were tested, the lipid peroxidation, reduced glutathione, and they established the antioxidant potential of white curcumin, as there was a significant decrease in the MDA level by 5.44 times, increase in glutathione level by 1.58 times, vis-a-vis the positive control group, and the level of GSH attained with TTO were, however, almost double as compared to the free white curcumin or tetrahydrocurcumin, indicating the antioxidant potential of white curcumin as compared to the naive or the free drug. The histopathological study revealed a complete healing of the skin on application of the THC-Tacro Ointment, and it confirmed the efficacy of developed TTO for the treatment of skin inflammation induced in animal model of AD. The molecular studies very clearly indicated that the levels, they were found to be similar as compared TNF alpha and interleukin 6 was similar to naive group and significantly different from that of positive group, indicating that our formulation could restore, it could have a very good anti-inflammatory activity, and it was similar to the naive group where there was no problem, there was no evidence of any inflammation. In summary, we can say that white curcumin nanoparticles, they were successfully prepared. They had very excellent parameters which were in range, whether we talk of particle size, zeta potential, total drug content, entrapment efficiency, and the particle size obtained was within the range, that is 109.4 as measured by the NTA. All the other studies clearly indicated the formulation stability and confirmed that the formulation, the lipidic nanoparticles, they have been aptly formed. SEM study showed clearly the morphology, and the rheology was very much in agreement with that of the shear thinning behavior. The occlusion factor for THC ointment was in comparison to marketed, and it was found to be stable at all temperatures. Stability indicated no aggregation. HPLC method was quite well in accordance to the guideline of ICH. No cytotoxicity was observed, and quite good quantitative, qualitative method for uptake were established, and the results were found to be similar to that of control cells when we observed it after 24 hours of treatment, showing that the formulation was quite safe. Thus, we wish to highlight here that the proposed THC-Tacro Ointment is a unique combination of concentrated lipidic nanoparticle, of white curcumin and ointment base and did not show any signs of inflammation. This is because of the targeted release, which was our main aim of formulating lipidic nanoparticle. That is the antioxidant at the site of action, which inhibited the tacrolimus-induced cutaneous irritation. So this was just a second part of the study, which we have already taken up. These are few of the publications from the work which we have done. The patent for this work has been granted and a part of this has been transferred to the company. The formulation will be transferred to another pharmaceutical company for scaling up and GMP manufacture will be taken up very soon. Thank you so much for your attention.